So the last video for the week, video number 4, is basically more about how human cloning might work. It actually could work if you were to utilize the Dolly method, and we have lots more methods and the approaches has been improved nowadays, especially with the discovery of CRISPR. But more or less true human cloning requires a somatic cells, as it says here in the second point. So somatic cells DNA is transferred, okay, or could be transferred to an enucleated egg cell because we do not want, okay, the properties from the donor egg cell. So we know that somatic cells are specialized cells, so direct transfer is of course impossible without it because it couldn't basically develop into other cell types. So some genes have to be turned back on before the process. So this is where it's going to be very complicated. You have to make sure that the genes could function later on. So human clone is almost an identical twin, but of course much younger than the actual human. So the original and the clone will have different fingerprints, even though everything comes from the human clone. So imagine if you were to clone yourself, your clone is you but very young in age. That is actually what happened to Dolly. Okay, so it's actually the same as the mother you, but it is younger in terms of its age. So let's have a look at the proposed approach, okay, to produce a baby. 3-parent proposed approach, okay, for using pronuclear transfer. So there was kind of like a rumor back in 2008 up until 2011 that a baby has been cloned, but it hasn't been more or less verified by a lot of people, so we don't know whether that's true or not. And I think starting from 2008, again, another news resurfaced of the creation of a human embryo or cloning of human embryo to produce a baby, but again it hasn't been more or less verified again. So we know that scientists, okay, whether they are mad or not, I guess they are still trying to come up, okay, with a method or approach, okay, to produce a clone baby regardless. But again, let's just get ahead, okay, and take a look at how, okay, they might do or produce a clone baby. So this is one of the approach that has been utilized and people are still trying to improve it. First and foremost, okay, you get a mother's egg. So over here is the explanation is that, okay, the mother's egg with abnormal mitochondria fertilized with sperm. So secondly, okay, you get another donated egg with normal mitochondria fertilized. So what will happen is that in the normal mitochondria and the abnormal mitochondria, you have to make sure this is a case of whereby when one parent has a disease that doesn't want to be passed on, again, to its baby or to the next generation. So zygote with abnormal mitochondria, what happens is that, okay, they just again remove, okay, the pronuclei, which means that it doesn't have the mitochondria that probably, okay, will affect or will pass on or will basically make the baby sick. And they transfer, okay, the pronuclei from the original parent into the pronuclei of the donor, which has been removed of its basically pronuclei or enucleated. Alright, so and then later on, similar approach to whereby they use electrofusion to make sure that the embryo is, well, might more or less go through mitosis to divide so that it produces embryo. And this embryo is now with normal mitochondria with the maternal and paternal genome transferred to the uterus, and then this could be implanted to the surrogate mother, either the original mother or another woman for surrogacy, and of course hopefully give birth to a child without the disease. Second approach is by the spindle transfer. So again, taking an example of a parent, okay, is affected with a certain disease. So of course, okay, an unfertilized mother's egg with the abnormal mitochondria with a spindle. So I'm not going to explain in detail what is a spindle. So if you have forgotten what is a spindle from your basic cell molecular biology or cellular biochemistry, you should look back, okay, on the spindle. So basically a spindle carries the DNA or genetic material as well. So again, you also need an unfertilized donated egg with no mitochondria. So the spindle and associated chromosomes removed from the mother's egg and transferred into the emptied donor egg, okay, removed of everything else, the pronucleus, apart from the mitochondria. So this is being used kind of like a case for the spindle and the chromosomes, okay, to grow into. So now with this case, okay, what makes it different, okay, from the first approach is that, okay, the reconstituted egg is then fertilized with the sperm of the father. In the first method, what happens is that uh, more or less fertilization doesn't really occur. Normally, it occurs in vitro, but over here, this is still in vitro. But then again, they try to more or less fertilize the sperm from the father before they electropuse it for further fertilization. This is one of the incorporation of another step. Okay, comparison between the two methods. So of course, again later on, once it fertilized, produce zygote that has more or less goes through mitosis, and this developed embryo could then be transferred to the surrogate mother, and of course, hopefully, give birth to a healthy baby without the disease affecting the parent. So, the science again in all this is that to make sure that for parents, especially with affected diseases that do not want to be passed on to the next generation, they could do that. But okay, making or keeping in mind again that they want their genetic material okay, to be more or less contained or kept, but without the disease. So this is one of the method, okay, the two methods that could be used. So they are making sure that their genetic material is being passed on without the disease, of course, but they also need another help from another donor in this case. Okay, so that's why this is a three-parent embryo to make sure that the baby is produced healthily, because we know and they know okay that there is a problem with one of the parent. Usually it's when it affects the female, because mitochondrial DNA is usually passed on to the babies, either the baby is going to be male or female. Now, let's have a look at some of the reasons, some of the myths, and something else again on human cloning. So let's take a look at the reasons first. So there are lots of reasons for cloning a human, or so to say. In terms of medical, so it is for curing certain diseases, as a revolution in cosmetic surgery, especially for plastic surgery. Organs transplantation would of course be useful. Cloning animal models of disease to study diseases further, especially in cancer research. Cloning stem cells for research, and also farming for drug production. Remember the farming from last chapter? Secondly, the reason is to cure infertility. So for those couples, okay, either the male or the female, okay, with fertility problem, may provide painless, inexpensive way for infertile couples. So far, it's actually not inexpensive. It's actually still quite expensive up until now. So the third reason, okay, this is actually done by a survey, so that's why I'm actually putting it there to prepare the parents. So to provide the best for the child. Number four, a step toward immortality. So that means that if you were to clone yourself, you are trying to keep yourself alive even after you are dead. You now, which is going to die, but you still have another you, your clone, going to be living even after you die. So some people thought that when you clone yourself, age is reversed, but that is actually not true. Okay, your clone is young, yes, okay, but you're not going to turn back. It's not a step, okay, towards fountain of youth. Who knows? Maybe someone will discover it, but that is not in the case of human clone. The next reason, financially secure family, especially if they are rich and probably they don't have a child. So of course they would want to make sure that all their assets, all their wealth, okay, is being kept in the family. So of course they would want to clone themselves or get a child by human cloning. The next one is saving endangered species. So think back of what I was saying earlier on. Okay, some species, some animals, those that are endangered now or extinct, probably have a good reason, or probably they have a good reason, okay, to clone them back. Okay, but for the woolly mammoth, just now, think about it. Is it worth it, or will it be a rampage in the world? Right. So the next one is better sense of identity. This is in case for a person probably with multiple personality. And don't take my word seriously, I'm just giving an example. So to discover your inner self, probably. Next one, special relationship among twins. Now, there are some people who are very envious of twins. I don't have a twin. I don't really want a twin. But I know a few people who has twins, because my cousins are twins and I have a few students who are twins themselves. So we know that twins, some of them, are close, and they actually have more meaningful relationship because they could understand one another better. They usually could sometimes finish each other's sentences. So some people would want to have those kind of relationship by cloning themselves. But again, bear in mind that your clone is younger than you, not exactly at the same age as you. In terms of DNA, yes. In terms of the actual age, no. And the last one here is request for cloning. So, as bizarre and as probably unsoundful as the idea was back then in the 90s, many people actually wanted to be cloned. So, they also want to clone a deceased child to make sure that the child that has died, okay, is being more or less, okay, reborn back. Now, let's have a look at some of the benefits of human cloning, and this is also based on the purpose, some of the good purposes, okay, for human cloning. For rejuvenation, reversing aging process. So to some people, probably if they were to create a clone for themselves, they can see their young self again, probably just to rejuvenate. Secondly, a breakthrough in human stem cells. Yes, that is actually one of the benefit of human cloning, to produce organs or tissues to repair or replace the damaged cells or organs. In terms of infertility, that is always good news. Gives hope to infertile couples to have children. In terms of reconstructive surgery, this is especially for those, okay, of accidents victims. Generation of bone, fat, connective tissue, and cartilage would help them probably store back their original self. In terms of defective genes, and this is related to gene therapy, which will be in next in line, okay, for this course, may be corrected, still ongoing. Down syndrome. So I'm not going to talk about Down syndrome, okay, but hopefully everyone knows, okay, what Down syndrome is. So Down syndrome is actually a common occurrence for those who are pregnant. They could actually test for if their baby is going to have a Down syndrome or not, starting from as early as 4 months. Uh, so some people would want to avoid the risk of having a child with a syndrome because probably they are not strong enough to go through life, okay, with a child, that because it's going to be challenging, right? And the next one is testing for genetic diseases apart from Down syndrome. There are lots of other diseases, especially those that could be passed on genetically. So the test may be useful and may be able to find a cure for genetic diseases in the future, in the hope. So now let's take a look at some of the risks of cloning. So many cloning experiments actually is not very successful. Animals or even the embryonic cloning of humans. Problems usually arise, okay, even from the successful clones. First and foremost, and this is going to be related to the dolly as well, high failure rates. So that is the number one problem. Incompatibility between inactivated egg and transferred nucleus always happens, even if they try to do the compatibility test beforehand. So successful transfer might not always ensure the egg will divide or develop properly. And embryo implantation as well into surrogate mother might also fail. So that's why, okay, they have to prepare a few embryos, okay, because if once they have transferred and pregnancy doesn't occur, they have to try again. So again, even if they became pregnant, pregnancy might also fail, may not come to full term, they may have miscarriage. Secondly, second problem, problems during further development if the first one has been overcome. So at birth, clone animals tend to be bigger than normal counterpart, referred to as LOS or large offspring syndrome. This is what happens to Dolly actually. Okay, so Dolly is actually born bigger than the actual you, and also normal you, a normal sheep. Okay, so loss is actually caused clones to have abnormally large organs, causing difficulty in breathing, blood flow, and etc. So even without loss, like in large offspring syndrome, clones might encounter kidney or brain malfunction or impaired immune system. How or why that occurs is still unknown and still being investigated. So the third one is on abnormal gene expression pattern. Okay, so the clones may look and have similar DNA sequence as the original human or the original animal, but genes might not express correctly at the correct time because it is a clone, but it is from a different time, so it has been reborn. So it will need complete programming to ensure normal or near normal development. And the last one is the telomeric differences. So this is what happened to Dolly. Okay, second one and the fourth one, telomeric differences. Again, I'm not going to talk about telomere. So telomere is basically a part of a chromosome, somewhere in the middle. So chromosomes commonly become shorter in time, okay, during cell division following aging. So as you age, your telomere will be shortened. So what happens with Dolly is that they actually took the DNA or from the chromosomes of a sheep, ewe, okay, which is actually about, I actually couldn't remember this, like about almost 10 years old of age, right? So when Dolly is born, so she is actually like it is actually about 10 years old. So plus 7 years that he lives, so it's about 17. So more or less similar timeline, okay, of the survival, okay, of a sheep. So chromosomes, okay, from cloned cattle or mice were found to be longer than normal and showed signs of youth as well as have extended lifespan. But this is what happens in Dolly, and that is why Dolly actually dies earlier. Dolly's chromosome was shorter than normal, which means that it's aging faster. So yeah, they did produce a clone of the animal, but then it doesn't live to a long age as they hoped it would be. So those are some of the risks. Now let's have a look, right, at the myth about human cloning. So some of the myths are a bit mind-boggling, but just bear with it. So some people thought, this is especially way before 2000, clones can be an instant, kind of like, okay, when you say that you want it, then it's there. No, it's not going to be like that. So, and they also thought that it appears at the same age as the original. So we know that that is not true, right, in case of Dolly. So it still has to be, even if you clone yourself, you are at 20 years of age, for example, your clone is still going to be a baby, 0 age, and then starts going to be 1, 2, 3, 4, and so more. Yes. Secondly, cloning xeroxes a person. No. Cloning in terms of xeroxing your DNA, yes. Xeroxing the person, no. This is nature versus nurture. Okay? Both have an impact. So nature would have an impact of how a person develops in terms of its personality. The same goes with nurture, the upbringing. So it may look and act exactly like the original, but it may have a different personality and whatnot. Right? Next one is cloning. Human cloning reduces biological diversity. I would tend to agree with this one because you are creating a clone with similar feature as you. So there is no variation. You are looking over here, looking over there, and everywhere there is you. Next point, clones would be less ensouled or subhuman than normal humans. In case of animal, we know that that is not true. Okay, so that means that they do have a soul, but whether they are less of an animal or subhuman than human is still not known. Clones could be used as spare organs for normal humans. Now that is not the function of human cloning, not to be sacrificed later on for organ transplantation. Next one, clones are created in batches. That is not true even for animals, unless it is for mice. Then that could be done, because it's not going to be easy to clone animal in batches, especially large animals and even us humans. So artificial wombs can be used to carry clone babies. So far, this hasn't been proven yet. If you happen to read over the internet, last year, maybe 2008 probably, but the news came 2019. They have created an artificial womb to more or less grow a lamb in it, and it grows to maturity. It's growing fine. But so far, there is no artificial womb to carry clone babies, especially for human. And the last one over here, human cloning is bad and is used only for bad purposes. I would say not really. It depends on, well, the intention of the person doing the cloning or producing the human clone. Alright, so I'll start here. On Tuesday, don't forget, again, we're going to discuss on Activity 1 and Activity 2. So if you have any queries on this week's lecture, prepare your question. We will discuss it on Tuesday. So I will give you the link to the meeting room. So till then, see you and stay safe.